ASRB Net 2024 preparation. This is preparation model paper 7, especially related to epidemiology. I'm Anand Shankar Reddy, PhD plant pathology. The first one is ability of a pathogen to cause the disease. These are all some kind of general definitions we might have studied in epidemiology, right? What is pathogen? What is sign? What is symptoms or such kind of things like pathogen, disease causing agent, the symptoms or external appearance of the pathogen infection or whatever it is, or sign or parts of a pathogen that we can see on the plants or such kind of things we might have studied, right? So now they're asking ability of a pathogen to cause the disease. So generally the ability of a pathogen to cause the disease is known as pathogenicity. So pathogenicity is the pathogen ability where it can cause the disease. So the second one is attachment of a pathogen on the surface of the on the host surface with the help of. They're asking how the pathogen will attach with the on the host surface. So to understand this, let's see, this is, imagine this is a surface of the leaf, and when the spore is landing on the leaf surface, so immediately, not immediately, by kind of signals or map and camp pathways. So whenever it perceives the signal, it starts to germinate by using a germination peg. This is called a germination tube. We can also call it germination tube, not peg. So from this germination tube, it will produce another structure called appressorium. This appressorium we use to helpful for the very strong attachment of the fungi. So maybe some kind of winds or drifts, maybe the spores may be washed out. So that's why it's produced another structure called appressorium, which very strongly holds or attaches to the surface of the plant. So this is generally called as attachment organ. From this appressorium, another, it will produce another structure called a penetration peg. The name itself indicates a peg-like structure where it penetrates inside. And another structure is produced from this penetration peg, that is called hastoria. Hastoria generally the nutrient absorbing organ of fungi. For example, you can take any organism. Generally energy will be required for the development or maybe for the growth, right? So here the nutrients are required for the growth and development of fungi, so that the nutrients can be absorbed through this specialized structure called hastoria. Since they have asked the attachment organ, so here there is a structure called appressorium. Appressorium is the attachment organ of fungi. Okay, the term hastoria was coined by Anton de Berry, whereas the term appressorium was coined by A.B. Frank in 1885. So generally the answer is appressorium. Appressorium is the attachment organ, whereas hastoриum is the nutrient absorbing organ of fungi. Next one, race specific resistance is otherwise called as. We'll understand something else first. So in epidemiology, we used to study two kinds of resistance. The first one is a vertical resistance. The second one is horizontal resistance. So vertical resistance, if I define like vertical resistance is a resistance that provide a complete resistance against very few races of pathogen. So it provides complete resistance, but only few races. It is exactly specific. For example, if you have a friend circle, that to very few friends circle, you are just in the specific in selection of your friends, that is called specific selection, right? So when it comes to horizontal resistance, the resistance that is actually that provides a complete not sorry partial resistance against a wide range of pathogens. Here, right, provides a complete resistance, sorry, partial resistance, but it operates against a wide range of pathogens. There is no specific selection. So that's why vertical resistance is called race specific, whereas horizontal resistance is called race non-specific resistance, because vertical resistance operates against only few races. It is actually specific, right? That's why it is called vertical resistance is known as race specific. The next one is examples of PR protein 5. What is PR protein? Pathogenicity related protein, right? So far Van and Loon, one of the article that was published by Van Loon in 2006 in annual review of pathology. We know very well annual review of pathology, one of the prestigious journal in India, that was established in 1963, right? So this Van Loon, his paper is suggested that around 17 families of PR proteins are identified, and a couple of articles in the recent articles 2021, one of the paper that was published, they have mentioned 19 PR proteins. There might be a chance also, because almost there are 15-20 years gap in between Van Loon and the recent publications, right? There might be a chance also, but still many of us are following that we have 17 PR proteins have been identified so far, that they are asking what is PR protein 5. First of all, what is PR protein? Pathogens related protein. We'll see how many classes of PR proteins are there first. So there are 17, as I told you, right? 17 families of PR proteins are identified. This is according to Van Loon. There might be updates also might be there. PR protein 1 antifungal, PR protein 2 beta 1 comma 3 glucanase, PR protein 3 chitinase type 1, 2, 4, 5, 6 and 7, and PR protein 4 chitinase 1 comma 2, PR protein 5 thematin like protein, okay, PR protein 6 proteinase inhibitor, they're all the 17 PR proteins. Everyone will find it. You just type something like the families of pathogens related layer protein comma Van Loon. You just type it, you will get all the 17 PR proteins. And the research article that was, I mean, review article that was published by Van Loon, so that you will get it again reading and all those things. Just time waste, okay, you will just go through all those things. You will come to know what the question they have asked. PR protein 5. What is PR protein 5? We have already saw PR protein 5 is a thematin like protein. So it is a thematin like protein. The next one is the tyloses are formed by. So what is tylosis? Just like some kind of blocking the vessels. So what vessels they are blocking, whether xylem vessels or phloem vessels, or what vessels they are blocking. Generally, tylosis can be observed mainly in the xylem vessels, formation of some kind of blocks in the xylem that can mainly lead to this tylosis, mainly observed in fusarium diseases. So generally it blocks xylem vessels. The next one is molecule or compound which elicits the defense reactions, that they are asking. HR, phytoalexin, phytoanticipin and elicitor. The answer was there in the question itself. They are asking which elicits or which activate the defensor. Elicitor. Elicitor is a compound that is produced by the pathogen, or I would say the pathogenic origin, like compound that is produced by the pathogen which actually elicits or which actually evokes the defense responses, that is called elicitors. Whereas actually pathogen also produce two kinds of things, elicitors and effectors. Elicitors are the molecule that actually triggers the plant defense mechanism, where the effectors are the molecules which actually you know suppresses some kind of defense, or may not get catched by the plant defense mechanism, so that it can escape and can cause, cause the infections. Of course, that effectors, we have a PR proteins. There are some kind of proteins, sorry, not PR proteins, resistant gene, I'm sorry. So these effectors were identified by some kind of resistance proteins. Around 8 to 9 classes were there, right? NBLRR, NBLPS, something, some kind of things will be there. Anyway, we are planning to do in a separate video, and especially on what is plant defense mechanism, host pathogen interactions, like what is the different kinds of molecules are involved in, like we are planning to do the molecular layers, what exactly, how this exactly the plant defense mechanism will work. Anyway, let's finish this first. The molecular compound that elicits plant defense is elicitor. Now the next one is the genetics and molecular basis of plant pathogenesis was written by, it is one of the book, a very important book that was written by J. van der Planck. First of all, discuss who is J. van der Planck. Is African scientist. We would say he's renowned as the father of plant disease epidemiology, and he also wrote many books. Around 5 books he wrote. Genetics and molecular basis of plant pathogenesis, defense kind of mechanisms, and various kinds of things he have written. Molecular basis of host pathogen interaction, and one of the famous book he was written by Plant Disease Epidemics and Control, which was published in 1963. Generally, this Van der Planck is considered father of plant disease epidemiology. So the book was also published by Van der Planck only. The next one, Indian stem rest rules, was written, right. There are two kinds of things which was related to wheat stem rest. Indian stem rest rules, Paksiniya pathway, both was given by Nagarajan only. But Indian stem rest rules was given by Nagarajan and Singh, whereas the Paksiniya pathway was given by Nagarajan and Joshi. The next question, Paksiniya pathway was identified by same Nagarajan and Joshi. Indian stem rest rules was given by Nagarajan and Singh, whereas Paksiniya pathway was given by Nagarajan and Joshi. So Indian stem rest rule 1975, this Paksiniya pathway probably 1980, I think. The next one is coconut lethal yellowing is originated from. Coconut lethal yellowing is originated from generally Cuba. Now we will see what are the different kinds of diseases that were actually originated and introduced into India. We'll see where it exactly originated first of all. These are all the list of important diseases, like late blight of potato, which was originated in Ireland, grapes, downy mildew, Europe, 45 to 50. Coffee rust Sri Lanka 1870, grapes downy mildew France 78, coconut lethal in Cuba 1904, chestnut blight USA 1904, citrus canker Asia, Brazil, USA and Africa. But citrus canker a little bit confusing. Sometimes people used to believe it's from China, and there are a couple of reports also available that is from India. And because the herbarium which was submitted by Indian government was earlier to this reports, and so many things will be there, but generally still the thing need to be sorted out. But it is generally now people are believing that it's Asia or some part of Brazil or US or Africa. Wheat stem rust generally originated in USA. Dutch elm disease US and Europe. Bunched up southern Wales. Banana sigatoka South America. Dutch elm disease Europe. Panama wilt Central Africa. Red of sugarcane India, Uttar Pradesh. Brown leaf spot of rice India. Wheat stem rust India, Madhya Pradesh. Tobacco blue mold Europe. Southern corn leaf blight USA. Apple scab India. And coffee rust Brazil. So these are all the list of important diseases that was actually originated and all those things. Since they have asked, so the answer of the question number 10 is Cuba. We have saw little things earlier, right? So coconut leather language originated from Cuba. So it's also important. We may expect years as well. And please take a look into. And monocyclic diseases are also otherwise called as. Monocyclic diseases are otherwise called as. Generally, monocyclic diseases are simple interest diseases. Polycyclic diseases are compound interest diseases, whereas polyetic diseases generally monocyclic diseases are simple interest diseases, whereas polycyclic diseases are compound interest diseases. Now, first of all, we will see this, and we have other slides to represent this monocyclic, polycyclic, and all the stuff. Anyway, monocyclic diseases are otherwise called as simple interest diseases. Generally, monocyclic diseases produce a curve called saturation curve. Polycyclic diseases are produced. Polycyclic disease, which curve they are producing? Generally, polycyclic diseases are known to produce a sigmoid curve, not sigmoid curve. We can also call it as a saturation curve. We can say saturation curve. So the answer is both B and C. Examples of polyetic diseases. Generally, citrus disease, virus, mycoplasma myellosis, Dutch elm disease, all are comes under the examples of polyetic disease. So the answer is all the above. Now we will see. These are all the some pattern of epidemics or epipoetics. The first one is monocyclic disease. Monocyclic disease produce a curve called saturation curve. Examples: smut, root rots, and wilts. Polycyclic disease produce a curve called sigmoid curve, also called as the S-shaped curve. And the generally diseases are like powdery mildews, rust, leaf spot, generally airborne disease. Like rice blast also airborne, right? This is lead blight of potato, generally is airborne. So these are all diseases are examples of polycyclic disease. They produce a curve called sigmoid curve or S-shaped curve. Polyetic disease, generally they produce a curve, bimodal curve. Examples are citrus virus, Dutch elm disease, fungal vascular wilts, mycoplasma myellosis. These are all the polyetic disease. First of all, I will tell you what is monocyclic disease. Monocyclic diseases are the diseases that they can finish life cycle, one life cycle in a in a cropping season. Poly means many. The pathogens which finish many number of life cycle in a single cropping season, whereas polyetic disease where it requires more than 1 year or 2 years to finish the life cycle. Okay. So monocyclic disease, generally the rate of inoculum is 0.02 units per day, whereas polycyclic is 0.1 to 0.5 units per day. Polyetic is 0.02 to 2.3 units per day. So sorry, per year, not a day, because they are polycyclic, right? Polyetic, sorry, polyetic disease. So these are all the some couple of patterns we do observe. And the next one is so vertifolia effect, 15th one. Vertifolia effect can be observed in. So what is vertifolia effect? First of all, we will see loss of horizontal resistance while breeding for vertical resistance. So if you are breeding, one resistance will be lost, and that is exactly horizontal resistance, while breeding for vertical resistance, that is called vertifolia effect. So the concept was given by J.E. van der Planck. We might have discussed in the previous slides, right? So this J.E. van der Planck is considered father of plant disease epidemiology. This vertifolia effect generally observed in late blight of potato. Okay, so the concept of inoculum potential was given by Van der Planck, S.D. Garrett, Nagarajan, and Baker and Cook. This the inoculum potential was given by S.D. Garrett. S.D. Garrett is considered as father of biocontrol agent related to pathology. But maybe entomology people may study Valentine really around 1870, 79 probably. But we do consider this S.D. Garrett is considered father of biocontrol agents. Which one is a simple interest diseases? Just now we have studied what simple interest disease, otherwise called as monocyclic. Compound interest disease known as polycyclic. Polyetic means there is no specific terminology, but generally monocyclic and polycyclic is considered as simple interest disease and compound interest diseases. So if they're asking the examples of simple interest disease, we might have saw the examples, right? Wheat loose mud, vascular wilt diseases, root rots, all are the examples of this simple interest diseases. Wheat stem rust is an example of simple interest, compound interest, both the and none of the. We have studied this wheat stem rust, the best example for compound interest diseases, right? The next one is polyetic diseases produces a curve is known as polyetic diseases produce a curve known as a bimodal curve. We have discussed it, right? So sigmoid curve, saturation curve, otherwise called as shaped curve. A sigmoid curve is monocyclic, polycyclic is a saturation or as shaped, whereas a polyetic produces a bimodal curve. So the 21 is phytopathometry used for. So phytopathometry generally used for plant pathometry. What is pathology? Study of plant is generally used for disease assessment. So for disease assessment we do use phytopathometry. The next one is San Prescott scale is developed for. So these are all some kind of scales, or I would say things that is actually developed for to identify a couple of kind of diseases. Generally, this San Prescott scale is actually developed for probably foliar diseases, I think. The next one is Uganda 99, UG99 strain reported for. So Uganda 99 is a strain that was first reported for wheat stem rust. I think everyone knows about this. Wheat stem rust, not stripe rust or leaf rust. So this was first reported in Uganda in 1999, whereas it was reported in India probably around 2008 in parts of Delhi, I think so. So the next one is Uganda 99 was first introduced in India, as the same question was asked. It's a Delhi, 2008 probably. In India, systemic analysis of pathogen races was started in the year of probably 1931 and 1932. I think 31, in the year 1931. The systemic analysis was the pathogen races was started. And the next one is world's first computer stimulation model used for forecasting of plant diseases. The first for stimulation model is Epidem. I think it was developed by Diamond and Wagner. Wagner and Horsfall, not Diamond and Wagner. Wagner and Horsfall around probably 1969. This is the first computer simulation model developed for wheat stripe rust or something else. Let's see in the later slides, I might have mentioned, I think so. Epidem was developed in the 1969 only, yes, no, I told it. Epidem was first developed by, just now we discussed Wagner and Horsfall, right? We got the same questions one by one, right? Okay, anyway, so this is the question. The first computer simulation model was Epidem, developed in the year 1969, and Epidem was developed by Diamond and Wagner. We will see the list of couple of forecasting simulation models. So these are also a couple of forecasting models like Epidem, early blight of potato. Oh, it's not stripe rust of wheat and all those things. Anyway, actually that's my mistake. It's been so many, so long, right? Anyway, so the Epidem is actually meant for early blight of potato and as well as tomato, that is Alternaria solani. Epime, southern corn leaf blight, Helminthosporium maedis. Epicon, southern corn leaf blight. Cercos, blight of celery. Mycos, blight of chrysanthemum. Epiven, apple scab. Epivet, viral disease of potato. Tomcast, early blight of tomato. Plasmo related to downwill of grapes, epidemic stripe rust of wheat. Anyway, that was actually I confused with this epidemic with the epidemic. That's why I told this epidemic is meant for stripe rust of wheat. Anyway, epidem is meant for early blight of potato and tomato, whereas epidemic is meant for stripe rust of wheat. Epipre, cereal rust and aphids. Blight cast, late blight of potato and tomato. Simcast, late blight of potato. Neckfry, late blight of potato. Plant plus Indo blight classed, Jugalsa classed. These all are developed for late blight of potato. And Blastiel, Epiblast, Pyricularia, Leaf Blast, Pyrenew, Blast classed. These are all developed for rice blast. SAS, strawberry diseases. Proplant, cereal diseases. Vegas, vegetable diseases. Cotton dog, cotton diseases. Epigram, generally related to barley powder mildew. These are all the couple of forecasting diseases. We may expect questions from this. The next one is boom and bust cycle was observed in. So first of all, we'll understand what is boom and bust cycle. So this boom and bust cycle was actually first given by Priestley around 1970. What this boom and bust cycle will say? So imagine if a new variety was introduced that is resistant to all the existing races of pathogen, immediately it got popularity because it is highly resistant. So due to the continuous usage, again the pathogen will break down the resistance, again can cause the disease. So when it gains the popularity, it is called a boom. Again, when the pathogen starts to infect, their popularity will go down, that is called buzz, boom and buzz. So this was first given by Priestley in 1970. Generally, this boom and buzz cycle was observed in vertical resistance. First forecasting model developed for late blight of potato, we have discussed, right? Blight cast. So blight cast is the first forecasting model that is developed. In the yesterday, see in the previous slides we might have seen this blight cast was first developed by probably scientist, actually forgot. In 1970, Cauchy et al. I think so. Yes, Cauchy et al. 1975 or maybe in between 70 to 75 probably. Anyway, I have given here. Yeah. Blightcast was first forecasting model that was observed in the year 1975. Okay, Cauchy et al. 1975 was the first forecasting model. Simulation model is Epidemiology, right? We have seen, right? The next one is Blightcast was developed by Cauchy et al. Just now we discussed it. Blightcast was developed by extended latency period observed in. So extended latency period can be observed in mentha rust. Agrobacterium transmits a gene through. So agrobacterium gene, generally agrobacterium known to transmit the genes through horizontal gene transfer, not vertical gene transfer. Active dispersal of plant pathogens is by, so active dispersal soil, seed. Okay, anyway, so these all are the examples of active dispersal of pathogens, not passive dispersal. Okay, so active dispersal through that happens through soil, seed, and as well as planting material. So the answer is all the above. Passive dispersal, of course, we know very well passive dispersal is through wind, water, insects, other kinds of things, that is called a passive dispersal. So the answer is all the above. So the next one is rain splashes mechanism of spore discharge occurs through Colotrichum lindimuthianum, gastricum, Puccinia regondata, and cladosporin. Generally, it spreads through air. Probably rain splash mechanism, Colotrichum. I think in a couple of Colotrichum, because it will produce, a depressed asexual fruiting body, we can call it as acerulate, right? Depressed pseudoparenchymatous tissues. So when the rain splashes sits on the leaf, so this acerulae, some spores it will produces, right? So there is a chance of spores that can split it out, and they can land on the other leaf surfaces or maybe somewhere else. So the rain splash mechanism of spore discharge, we can also, of course, other diseases also might be there, but among the given options it is probably Colotrichum lindimuthianum. Generally Colotrichum, you can remember. And the next one is a race concept is not applicable for the following: Puccinia, Sclerotinia, Fusarium, and Firuclara. Probably it's Sclerotinia, because there is no such kind of races system will be there in Sclerotinia, right? Maybe one or two species will be there. Mostly Sclerotinia, Sclerotiarum, Sclerosium ralph. This one or two species. There is no much variations can be observed in this Sclerotinia, Sclerosium ralph, and all those things. But we can see Puccinia, Fusarium, Firuclara. We have hundreds of races like Puccinia graminis stratici, Puccinia graminis hardy, Fusarium oxysporum, pharma species, Vasenpectum, cubins, Pyracularia, varese, and various things will be there, right? So when it comes to Sclerotinia, there is no such variation can be observed. Probably the answer is Sclerotinia. The next one is rain tap and puffing mechanism of spore discharge can be observed in. Rain tap and puffing mechanism, Lycopodon, Geostrum, Puccinia regondata, and Cladosporium. Probably I was a little bit doubted about this. I have no idea regarding this. Anyway, please, you guys, if you know, please let me come to know. The next one is missed pickup mechanism of spore discharge can be observed in. So missed pickup generally is an example of Cladosporium. The next one is domestic quarantine is a total failure in which country? Domestic quarantine is a great failure in India, because we have lots of diseases, and that are actually spreading through borders to borders, because our border quarantine, that is a domestic quarantine, is not at all strict, right? That's why in India domestic quarantine is a complete failure. There might be very other reasons also might be there. Then you can ask me, in which country the domestic quarantine is very strict? In which country the domestic quarantine is very strict? Best example I would say, in India the domestic quarantine is very, 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 very strict. Sorry, In Australia, domestic quarantine is very strict. It is one of the toughest country where domestic quarantine is going is very well. The next one is which path gives the best coverage of field for a systemic sampling. So if you want to go for systemic sampling, there is a method called W-shaped methoding. So this W-shaped methoding that gives the perfect coverage to get the samples and all those things. So this method is W-shaped methoding. Next one is epidemic is meant for, I think we have already discussed, epidemic is meant for early blight of tomato and potato. Epidemic is a forecasting model developed for, we have already seen anyway, wheat stripe rust. Plasmo represents for plasma parviticola, that is grapes downy mildew. Dutch rules are applicable for, so there are a couple of rules like Dutch rules, Van Everdingen rules, Irish rules, all the rules are applicable for one disease, that is a late blight of potato. But Mills rule is different. Mills rule is meant for apple scab. That was developed by Mills probably in 1944, I think so. So Mills rules are applicable for apple scab, that is sure. First expert system that is developed against 17 soybean diseases. What is that first expert system that they're asking? It is a very famous one that was developed by US in 1983. So that expert system name is Plant DS. I will show you a couple of other expert system on the later slide. The next one is coconut lethal yellowing is transmitted by coconut lethal yellowing is known to be transmitted by leaf hopper. I think a Mendes Clodas, I think so. Now these are all a couple of expert system. The first one is a plant DS developed for 17 soybean diseases by USA in 1983, as I told you, right? Pome, that is developed for apple, potatoes, early blight of potato. Tomex, which is equipped with 78 questions and 87 photographs for diagnosing around 37 diseases of tomato. German, this is related to greenhouses, and Proplant, Epipre, Exis, Tomex UFE, and Simfed 3 and SACO. These are all the various kinds of expert systems that has developed. But the first expert system is Plant DS that was developed by USA for soybean disease, around 17 soybean diseases. Then you can ask me what is expert system. It is actually a computerized model, with having a problem ability, solving skills with a human expert or human help. With the help of human, it can solve the problems. The next one is who written Plant Disease Epidemics and Control. As I told you, right, J. Vanderplank wrote 5 books. Among the 5 books, this book is very, very, very famous. Plant Disease Epidemics and Control, which was written by J. Vanderplank in 1963. So the answer is J. Vanderplank. Dutch rules are formulated by. So they're asking probably Van Everdingen rules, Mills, Beeman rules, all are Van Everdingen rules and Beeman rules are late blight of potato. They have different rules. Mills rules are Apisca. Probably the answer is La Plante. So the La Plante was the one who formulated this Dutch rules. So Beeman period is related to. This was one of continuously asked question. Not in it, but Dutch rules, or otherwise called just a minute. The fifth question is Dutch rules, right? So Dutch rules are also known as Van Everdingen rules, I think so. Dutch rules are otherwise called as Van Everdingen rules. Probably the answer is Van Everdingen. Anyway, a little bit confused about this question. Anyway, you guys know, please let me tell. So I believe is it's Van Everdingen because Dutch rules are also known as Van Everdingen rules. That's right. Okay. Beemont period is related to late blight of potato because in Beemont, which was there in between the period of around 1850s and 1850 to 53 probably. So the late blight also happens in 1845 to 1852, right? So this Beemont is actually one who also formulated Beemont rules for the identification of this late blight of potato. So this period is actually related to this late blight of potato. The next one is the chances of epidemic development is less in case of the pathogens if they are carried out by seeds, vectors, water, wind. So seeds. So the chances of epidemics are very less if carried out by seed compared to other. So plant disease can be measured by using disease incidence, disease intensity. Anyway, plant disease can be measured by all the above, right? We used to measure by using disease incidence, severity, yield loss, and all those things. So the answer is all the above. PLAM is a computerized prediction system recommended for. So in the computerized models, we have already seen the PLAM is actually groundnut related diseases. I think groundnut leaf spots related diseases. And the first scale for estimating the plant diseases was developed by ICAR is the one who developed this a scale for estimating this plant disease. NACOP is known as considered as father of American nematology, right? Epidemiology generally deals with survival and spread of pathogens, morphology of pathogens, reproduction of pathogens, suitability of general answer is survival and spread of pathogens, environmental factors, how the pathogens survive and spread, all those things that was dealt by epidemiology. And J. Van der Planck is considered the father of plant disease epidemiology. When the primary inoculum is responsible for creating the epidemics from outside the affected area is called as, so it is coming outside. If it is inside, it is called endogenous epidemics. If it is coming outside, it is called exogenous epidemics. Disease comes under endogenous epidemics. What are the examples of disease that comes under endogenous epidemics? They are asking. For example, rice blast, wheat stem rust, grapes downy mildew, none of the above. So rice blast, best example of endogenic is rice blast, because the wheat stem general spores come from outside, right? Grapes downy mildew, soil-borne pathogen. The next one is of course the sporangia also can spread through air for a couple of little bit distances, but generally the answer is rice blast. Diseases comes under exogenous epidemics. What are the diseases that comes under exogenous? Best example, wheat stem rust, just now we discussed, right? Which pathogen causes severe epidemics? Airborne pathogens causes destructive epidemics. Generally they spread through air and spread very fast, so that it can cause epidemics. Introduction of variety TN1 in Tamil Nadu is associated with the epidemic. This TN1 is a very famous, that is actually associated with a disease called rice bacterial leaf blight, which was severe in first it was very severe in probably Bihar 1963, and later it became very severe around 1980s and 1990s in Tamil Nadu because of the introduction of this variety called TN1. Artificial epidemics created for experiments are generally called as, what we can call it as, progressive epidemics, field epidemics, isolated epidemics, endogenous epidemics. Then called as field epidemics because we are creating artificially, right? So the next one is Dutch elm disease, bacterial wilt of cucurbits and pine wilt disease are primarily disseminated by. So Dutch elm diseases are known to be transmitted by beetles, but bacterial wilt of cucumber also beetles, that is corn flea beetle and all those things, Diabrotica victoria, Diabrotica duodenum punctate, we might have studied, but pine wilt I have no idea. Since the two options I found it is a beetle, so probably the answer is beetles compared to the leafhoppers, plant bugs and mealybugs. So the answer is beetles. Disease epidemic growth is which process? So they're asking disease epidemic growth is a which process? Spatial, temporal, both A and B, none of the, probably it's both spatial and temporal, I think so. The next one is the key factors for the prediction of late blight of potato. Temperature, moisture, humidity, both A and B, I think both the temperature and the moisture are the important factors for the prediction of late blight of potato. Uganda 99 is a strain of, you know very well, that is a strain of wheat stem rust, which is caused by Paxina gramineous. Delhi declaration of wheat stem rust is Uganda 19 adopted, which happened with, we already discussed, right, 2008. So Uganda 99 means it was first reported in Uganda 1999, but it was first reported in India, Delhi in 2008. So the answer is 2008. Next one is the growth curve of development of epidemics. Sigmoid curve, linear curve, saturation curve, and a L-shaped curve. It is the growth curve generally a sigmoid curve. Which of the following are decision support systems? Wisdom, radar, POWs, all the above. Generally, all are the support system. The next one is the leaf wetness period that is required for the initiation of apple scab disease. Generally, the leaf wetness period may varies, depends on the temperature. So the temperature is also one of the factor where it affects this leaf wetness period. So generally, it is believed that apple scab, the leaf wetness period that is required for the initiation of infection is around 9 hours. You can remember apple, APPLE, scab, SCAB. Apple, how many letters? 5 letters. Scab, how many letters? 4 letters. 5 plus 9 is equal to 9 letters, which is 9 hours. You can remember in that way. That is also one of the method, right? So that is generally it is 9 hours. The next one is the terminal velocity of the spore after takeoff is depends on. So the terminal velocity, size, shape, appendage, I think probably all the above answer. I'm not sure, probably all the above. Disease favored by the acidic soils. Which of the diseases are highly favored by the acidic soil? Wilt of pea, wilt of potato, viticulture disease, potato scab. Acidic soil, probably wilt of potato. Potato scab is severe at saline salat 8.0 wilt of viticulture. Probably it's a wilt of tomato. If it was there in club root of cabbage, so the answer is club root of cabbage. Since there is no such options, probably it's a wilt of potato. Disease is favored by alkaline soils. Alkaline potato common scab, just now we discussed, right? Potato common scab is highly severe at alkaline soil. Leaf fall of rubber is caused by. Rubber leaf fall is generally known to be caused by Phytophthora palmivora. Uckelberg's apparatus is used to determine terminal velocity of the spore, weight of the spore, height of the spore, size of the spore. Generally, Uckelberg apparatus is especially meant for the terminal velocity, at what velocity it is traveling. The terminal velocity of the spore initially, so that is used to identify by using an apparatus called Uckelberg apparatus. System analysis of brown rust of wheat in India was system analysis, or what? Systemic analysis. Probably there might be a little bit typographical error as well. Probably it may be systemic analysis, I think so. Anyway, brown rust of wheat was first proposed in India by Nagarajan et al., who first demonstrated that disease resistance is inherited by a characters Biffen. 1905, probably through Mendelian fashion the diseases are spread. That was first given by this Biffen. This is also one of the very good discoveries related to how the resistance, like at the time scientists used to believe that every time we need to create resistance. So when the Biffen discovered first time in 1905, so they identified that resistance also can carry it along with the next generation to generation. That was first discovered by Biffen, Orton and Biffen, 1905. Who developed a method of predicting wheat stem rust pathogen by using a satellite television cloud photography. Nagarajan and Singh only. That's why how they discovered this Paxenia pathway wheat stem rust also. Okay. The next one is Apple scab warning services was developed by Gupta et al. In the following. Generally, it was first discovered in Himachal Pradesh. It is also one of the highest apple growing areas, right? So Himachal Pradesh. Late blight of potato forecasting model. So which of the following is a late blight of potato forecasting model? Dutch rules, Beeman rules, Wallin and Homans rules, all the above. Probably the answer is all the above, because I know this Dutch rules and Beeman's rules are especially meant for late blight of potato. So since we cannot say only Dutch rules or maybe Beeman rules, so probably so we can think in this way also, so that we will get the answer. I never heard of this Wallins and Heyman rules. Probably there might be related to late blight of potato, but this Dutch rules and Beeman rules are especially meant for late blight of potato, so probably the answer is late blight. I mean all the above. The next one is potential ascospore dosage, generally called as PAD, is an important component in forecasting of which diseases? Rice blast, white rust of crucifers, apple scab, sporangia. Rice blast, white rust of crucifers, no, all the above. Probably it's apple scab. I think I have heard regarding this potential ascospore dosage related to apple scab only, I think so. Probably, I'm not sure. Probably apple scab. Erickson developed a concept called pharmospecies through cross inoculation with what? Cross inoculation with wheat stem rust only, right? Which of the following rust iridospore is not designated for long distance spread? They're asking which iridospore is not designated for long distance spread. Generally in wheat stem rust we know this rust is known as this iridospore is known as summer spore and long distance spore and all those things we might have studied, right? So in one rust that is called leaf rust, it is reported that this iridospore cannot designate or designed for a long distance spread. Maybe probably teleospore may spread, probably, but in stem rust, wheat stem rust, of course this iridospore and teleospore can travel. And disease equal to inoculum into, this is like some kind of thing, disease equal to inoculum into disease potential. Very simple. And epidemic rate curves for the late blight of potato. Symmetrical, bell-shaped, both A and B, asymmetrical, probably both A and B, symmetrical and bell-shaped, both epidemic curves of late blight of potato. And the next one is fourth and fifth factor added to the disease triangle. What are all the fourth and fifth factor? They are asking time and human. Three factors are there, like host, pathogen and environment. Fifth factor is time, and sixth factor is human, right? It all puts together called a disease pyramid. Area under disease progress curve, that actually refers to what it actually refers to: area between the start and end point, the area covering the area between the disease progress x-axis on the graph. This is generally answer is C. So area under disease progress curve, generally that actually gives like some kind of axis. So probably the answer is between the progress curve and the x-axis of the graph. Basidospores of stem rust of wheat can attack wheat, barberry, both A and B, Thalictrum species. See, this basidospore only attack barberry. It cannot germinate on wheat. Iridospores and teleospores only grow on wheat. Basidospores, Asium and Pycnium, Asium, Basidios only germinate on barberry. Even though you put teleospores and basidospores on the barberry, barberry cannot give the symptoms. Even though you put basidospores on the wheat, there is no germination because it's not compatible that way. So the basidospores of wheat stem rust only attack barberry but not wheat. Protein for protein hypothesis was proposed by protein for protein hypothesis. I think Vanderplank proposed as protein for protein hypothesis. Gene for gene hypothesis, H.H. Floor, we know very well, 1946, right? So when energy for spore liberation comes from an external source, such a spore dispersal is known as, I repeat, when energy for a spore comes from an external source, such a spore dispersal is known as, of course, passive dispersal, not active dispersal, right? It is coming from external sources. That's what it's called as passive. The next one is factors that influence the incubation period. What are all the factors that influence host pathogen? Of course, all the above, right? Host pathogen environment all will affect the incubation period. The crop yield with no disease measures imposed are known as crop yield with no disease measures imposed, right? Attenuation period, economic yield, theoretical yield, primitive yield. I don't know primitive yield, like probably it's a primitive yield. I think no disease measures are imposed. Probably it's a primitive yield, not economic yield. So probably, I'm not sure. Please let me know about 92. Okay. So measures of plant diseases intensively is generally done in terms of yield loss, incidence, severity. Measurement of plant disease, of course, we have already discussed it. So answer is all the above. We can measure by using yield losses or the percentage of incidence or the severity. Okay. So blossom blight represents for blossom blight of apple. We have discussed earlier, right? The reproductive success of a pathogen under a wide range of conditions is known as fitness. The blight rate, the highest rate of epidemic development, generally occurs in a vegetative propagative crops followed by self-pollinated crops or cross-pollinated crops is mainly due to the highest rate of epidemic development generally occurs through in a vegetative propagated. Okay, vegetative propagated followed by self-pollinated crops and cross-pollinated crops is mainly due to it's a gene uniformity or genetic uniformity. Okay, gene for gene hypothesis represents, I will tell you without seeing the, for every resistant gene in the host there must be a corresponding and avirulence gene in the pathogen. For every virulence gene in the host there must be a avirulence gene in the pathogen and vice versa, like that. That is called gene for gene hypothesis. First was given by H.H. Floor 1946, right? Which model is used to see the relationship between yield loss and assessment made at several times in a cropping stage is generally known as a multiple point model. In the multiple point model only where we see the relationship between the yield losses and assessment made. Uganda 99 is a strain of a wheat stem rust that was first discovered in 1990 from Uganda. That's why it's called UG99. Okay. Which model indicates the statistical relationship between host and a pathogen at a specific point of time is known as critical point model. There are a couple of models like multiple point model, critical point model, and other models also there. So generally the model statistical relationship that we can observe only critical model. We can see the some balancing points so that we can understand this. So critical point model this is. So these are all the answers related to. So this is my book, A Vision into Plant Pathology, complete student version. This might be useful for you during your preparation, not only for NET but also for JRF, SRF, or whatever the exam you want to prepare. This will be definitely helpful. Doubts or clarifications or for further guidance, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.